Hi, I'm Maha, and we're going to be talking about Southern blot. The following diagram is an overview of Southern blot analysis. We will be going through each of these steps in more detail. If you would like, pause and take a look. So firstly, in DNA extraction, the DNA source, which can be obtained from tissue cells, blood cells, and so forth, the cells are lysed via SDS detergent, the proteins are degraded, and the DNA is isolated and dissolved in a tris buffer or water. So next is restriction enzyme digestion. So the isolated DNA is digested with restriction endonucleases, which cleave DNA at a specific sequence, like the image shown above. Following restriction enzyme digestion, these DNA fragments are run through a gel electrophoresis gel. These DNA fragments are all negatively charged and will move towards the positive end of the electrode, and they will be separated by their molecular weight. Next, the gel is transferred onto a filter. The gel is placed onto a paper wick and a connection is formed between the gel and the reservoir. A positively charged nylon membrane is placed on top, covered with paper towels and held by a weight. Capillary action allows the buffer to soak through the filter and the DNA fragments are immobilized onto the membrane. DNA fragments on the nylon membrane are hybridized with radioactively or fluorescently labeled probes that bind to complementary DNA fragments of interest. It is then exposed to an X-ray film which records regions of radioactive decay, and an audio radiograph is obtained. There are advantages and disadvantages to the use of the Southern blot. It is relatively quantitative and useful for the study of longer fragments of DNA and can be used to detect homologous genes in the genome. However, it requires a large amount of source DNA and for this reason has been replaced by PCR. Southern blot is used in support of restriction fragment length polymorphism studies. Since different individuals in a population carry a variety of point mutations in their genome, these can alter restriction enzyme recognition sites, so when treated with restriction enzymes, the same set of fragments are not obtained. Thank you so much for taking your time to listen.